Can you hear me?
Yes.
Okay.
No
Okay.
problem,
Thank you very
yes.
much. Thank you for a very nice introduction. I'm very happy to talk to colleagues in Turkey. That some of you we meet every 2 years in the IMIG, and hope to meet you soon in person. This time it will be a virtual presentation, and the topic of today is about biomarkers, and a subtitle is Inherited Genetic Mutations, Polymorphisms, Prognostic and Predictive Biomarkers. Let me see. Yeah, I have no disclosures. Just to tell you where I'm now, I'm in the big red, under the big red arrow is Trondheim. We're right under the polar circle. It is the third city of Norway. It's a university city with 30,000 students. It's called NTNU, Norwegian University of Science and Technology. It is actually the biggest university in Norway at the moment. Here are the some of the main topics: mesothelioma, epidemiology, pathology, prognostic biomarkers, predictive biomarkers, BAP1, genetic mutations, DNA repair and other genes, single nucleotide polymorphisms, and conclusions and future perspectives. Just for the history, asbestos has been used for thousands of years, and here is an example from archaeology in Norway. These are pottery with asbestos clay that people used to make food inside. Quite big pottery, very nice. Some symbols around, but it was actually a very popular material for a long time. But the big problem started with this man that invented Eternit. He patented the combination of asbestos, chrysotile asbestos with cement, and he made these very famous roof material and other materials. Then started the big production, and then started also the big problem with mesothelioma. And here we see the typical mesothelioma mine, which is an open mine. And here is in Cyprus. There is a big mine in the Mount Troodos on the Greek side. It is, you can see it's a little lake in the middle. And the whole thing has been open before. Now they have made some woods, some plants to keep the asbestos down, but still it's a lot that is out in open air. But still people they have made this to a park, and people go there for picnic, and I don't think that's a good idea with asbestos all around, you know. Just a few more slides on asbestos and epidemiology. Here you see that the consumption of asbestos was enormous from 1920, with a maximum in 1975-80, and then going down, but still there is a lot of asbestos produced in many countries. Here is in the Nordic countries: Norway in blue, Denmark in red, and Sweden green. And we see that Norway had the smallest consumption. And that's why Denmark has a bigger problem than Norway. Denmark consumed much more asbestos and longer than Norway. And we see that in North Denmark, where we have the asbestos industry, they have still a big increase in asbestos in mesothelioma cases. So, as you know, most of you know about this, but it is one of the most important occupational carcinogens and cause about half of the deaths from occupational cancer. So it is the inhalation of the fibers that is dangerous, and we know that there are more than 125 million people exposed at the workplace, and more than 107,000 people die each year from asbestos-related disease. So these are the diseases that are connected to asbestos. We see it's not only mesothelioma and lung cancer, there are also laryngeal cancer, ovarian cancer, and also asbestosis. Asbestos used is chrysotile. This was this man in 1960 who made the association between asbestos and mesothelioma in South Africa, Dr. Wagner, which this is like a very, very important article and a very interesting article to read. If you haven't read it, I would really read it, because he not only finds the connection of occupational asbestos, but also environmental asbestos. Everything is in this article. It's very interesting. What is also interesting is that mesothelioma starts in the thoracic parietal pleura, and not in the lung pleura, not in the visceral pleura. It's just when it starts spreading, it goes to the lung pleura, and then it suffocates the lung, and you can see this thing happen. And how does it affect the mesothelium? It actually can both indirectly make an inflammation, but it can also directly spear. Here you can see an asbestos fiber that spears the nucleus and damages the mitosis. There are several molecular changes from asbestos. I will not go in detail, but there is a lot of literature on this, and we have written in 2015 we published a review where we have gone a little more in detail on these molecular effects. When we look at his histopathological subtypes, there are three main subtypes: epithelioid, sarcomatoid, biphasic. The epithelioid has the best survival with a median of over 12 months. The sarcomatoid less than 6 months. What is important is that in the last 40 years, maybe, the long-term survival has not changed. If you see here, 1960 to 79, 80, 90, 2000s, the curves are almost overlapping. So we haven't been able to change the survival significantly, at least not the long-term survival. The prognostic factors, this is the strongest prognostic factors, the epithelial versus non-epithelial types. Female has a better prognosis, younger have better prognosis, but still the differences are in months, it's not years usually. We have just published a paper, I don't have the reference here, but we found that in patients treated with any therapy, only the histological subtype was a significant prognostic factor. not age, not sex, not comorbidity, only histological subtype. That means that we don't need to be so afraid to treat somebody with epithelioid or an epithelioid. We should try to be more aggressive with epithelioid types to treat them, and those with the sarcomatoid, maybe we should be less aggressive to treat because they have a very bad prognosis anyway. But this may change very soon. Biomarkers. The probably most important biomarker for mesothelioma is mesothelin. It was discovered in 1992, and it's highly expressed on the cell surface of mesothelioma, but also many other cancers like pancreas, lung, stomach, triple negative breast cancer. It's expressed in 30% of human cancers, and it is a possible target for immunotherapy or antibody therapy or other therapies, and probably CAR T therapy. Mesothelin is also elevated in the blood and pleural fluid in half of mesotheliomas. It is not elevated in the blood in other cancers. This is important. This is a very important study by Robinson and his group in Perth, published in the Lancet 2005, where mesothelin in the serum was elevated in mesothelioma, but not in other lung-related diseases. One moment. Έλα τώρα, δίνω διάλεξη, θα τα πούμε. Εντάξει? Yeah. So, one thought that this could be a very good biomarker for mesothelioma, a circulating biomarker, but only 50% have elevated mesothelin. And we see that in some cases, they had an increase in mesothelin before diagnosis. We also see that we have a change with chemotherapy as well. This is not so very good, but this is maybe better. We see that there is a fall in mesothelin after chemotherapy at response, and it increases at progress. There are several other biomarkers that have been tested, like the ENOX2. This was seen upon as a very promising biomarker, as it was increased in blood 4 to 10 years in advance of clinical symptoms, but it has not been validated. We see now other tests coming, like for the cancer-seq, which is a combination of mutations and proteins, which is a multi-cancer test for early detection, but we see that mesothelioma is not here. And it's not a good test for lung or for breast. It's very good for ovary and liver. We have a study called Cancer Biomarkers in Hunt since 2013 to identify biomarkers in serum, early biomarkers for lung cancer and mesothelioma in an early stage or early detection. And we have tried to combine it with a clinical model. So this is the research center, which is outside of Trondheim. It is a big biobank of more than 120,000 people's blood, and a lot of clinical information, maybe 300-400 variables per person, and it's follow-up for 20 years. So it's a very big biobank, and we use material from this biobank. And already we have some candidate markers with metabolomics for mesothelioma diagnosis 1 to 16 years before diagnosis that we are working on right now, and we are trying to validate. We have not validated, but we have some interesting markers. We also have microRNAs where we can use signatures to detect mesothelioma. This is also ongoing project. And also proteomics, where we have signatures. For example, one signatures with 11 features, where we can separate mesothelioma, which are the red, from the controls. This is 1 to 16 years before diagnosis. They're very interesting ongoing studies, no validation yet. A little bit about predictive markers. Here is the study that started the treatment of pemetrexed and cisplatin, the Vogelsang study, which has been our basic treatment for mesothelioma for so many years. But there are no biomarkers. We don't know who is the responder and who is not a responder, because maybe 50% are resistant. We don't know who are the responders. Then came the treatment with bevacizumab, which is good, but also there we have no biomarkers. Half of the patients are not responding. How can we select? Then came the double immunotherapy with Nivo and IPI in the presidential symposium last year, with a very nice survival curve. And we can see that it is significant better than the epithelioid, but not so much better, a little bit better, but it's much better for the sarcomatoid. So if you put the curve of the sarcomatoid, it almost goes over the curve over epithelioid. So actually, here we have a treatment that is effective for the sarcomatoid subtype. And this is a great news for sarcomatoid tumors. But still, we don't have any biomarkers also for this. We don't, PD1, PDL1 is not biomarker, tumor, also TMB is not a biomarker. So for mesothelioma, we have no biomarkers for any therapy. The only actually biomarker is sarcomatoid for double immunotherapy. Yeah, so this is where we are today. And it's also, of course, it has been FDA approved. But now comes slowly new biomarkers for immunotherapy. And here is a study from Mankor and et al. From eBiomedicine, showing that CD8 T cells in the blood that re-expressed CD45RA, TEMRA, they had a high survival. And this may be the first biomarker for the combination of IPI-NIVO. It's a very interesting study, and it really shows a difference here. So I will go now over to the genetic component why do we suspect there is a genetic component in mesothelioma? We have many examples of people with heavy exposure that never get mesothelioma and people with very low exposure that get mesothelioma. So probably those who have a heavy exposure and don't get mesothelioma, they have a very a good system of reparation, because we know that asbestos makes damage. Yeah. So as all of you know, in 2011, the BAP1 was described as a mesothelioma susceptibility gene. And it is a cancer syndrome that not only mesothelioma, but also uveal melanoma and melanocytic tumors and renal cancer, and other cancers as well. And BAP1 was proposed as a novel target for prevention and early detection from Carbone in 2013. It was striking that in 2 families with BAP1 mutation, from 28 persons, 12 had mesothelioma. So almost half of them got mesothelioma, and they also had other cancers. But they were not asbestos exposed. So the question is this mutation also a susceptibility for asbestos exposed mesotheliomas? And there are some mouse studies that show that yes, if you are exposed to asbestos and have the mutation, you have a much higher risk to get mesothelioma. That's for mouse in mouse, okay? I will not go into this. What is the role of BAP1? BAP1 functions as a deubiquitinase, and it affects the BRCA1 BARD axis with DNA repair. It affects cellular differentiation, and it affects proliferation and cell cycle regulation. So it is a very broad effect protein. So if BAP1 is not there, many mechanisms are in trouble. What is interesting with this familial BAP1 syndrome this is a busy slide, I know, but if you look at, for example, the cutaneous melanoma, the clear cell carcinoma, the meningioma, with BAP1 mutation, they have very bad prognosis. The mesothelioma is the opposite. If you have a BAP1 mutated mesothelioma germline, then you have a very good prognosis, which is exactly the opposite. And this is a very interesting point that gave us some ideas. But which are the germline mutations and how common are they in mesothelioma? My PhD student then, Vasily Kipano, went to Chicago and she stayed with the group there and is the first author of this paper, which reports the frequency of germline mutations in malignant mesothelioma. They extracted from saliva or peripheral blood the germline variants. They sequenced tumors, and they looked at 85 cancer susceptibility genes. Oops, yeah. So here you can see that all the genes that were mutated, BAP1, BRCA2, CHEK2, CDKNA2, ATM, BRCA1, and others. So 24 germline mutations in 13 different genes in 23 patients. So 12% of all mesothelioma patients had the germline mutation. Of course, this was a selected group of people in a highly specialized center in America. Probably the number, if you look at other centers, will be lower, but it is a high number. I will not go into this. I will not go into this. What is interesting, though, is that some of the other mutations, most of them are in the DNA repair mechanism, but some of them are in chromatin modification genes, like this SWI SNF complex gene. And these genes are mutated in 20% of all human cancer types. So it is very, very common and very basic mechanisms, genes that are mutated in mesothelioma. But then the survival was so high in these germline mutated mesotheliomas, was 7 times longer than wild type mesotheliomas. And they even had other cancer types, but they still lived much longer, as you can see these survival curves. And the researchers were thinking, okay, this is a prognostic marker. This is a prognostic marker. They have a more a good biology, not so aggressive. Well, we wanted to see whether this is a predictive marker, and we published as an abstract last ERS Congress that BAP1 loss predicts longer survival for mesothelioma patients treated with pemetrexed-based chemotherapy. And we combined our patients in Denmark with with patients in Perth, some patients in Perth. And if you see here, the left curve are those treated with pemetrexed platinum. The right curve are those treated with best supportive care, no chemotherapy, okay? On the left, we see the BAP loss patients in blue and the BAP retained patients in red. So we see that the BAP loss patients, they have a median survival of 20 months versus 10 months in the others. And if you see the survival in the best supportive care group, it is 10.5 and 4, but it's not significant difference. You see that the curves are just separating a little bit in the middle, but then they go together again. So it's actually no difference. So that means that BAP1 is not a prognostic factor. It is a predictive factor. So we believe that this is the first clinically relevant biomarker for chemotherapy in mesothelioma. And we are now looking to see whether it can be a predictive factor for immunotherapy and for immunochemotherapy in mesothelioma. So we are starting a larger project with other countries to try to find out if this is true. Okay, I have to go back to show you this. What is a detail? It's not shown here, but I can tell you that sarcomatoid patients with sarcomatoid tumor with BAP1 loss had the same effect of chemotherapy like the epithelioid. Okay, so this is a very, very interesting detail. We also wrote with Vasiliki, my previous excellent PhD, and now she already attendant doctor at Odense Hospital, University Hospital. This is a review on the mutations and polymorphisms. It's open access. You can download it. We went through many papers, and we found that for germline changes, we found that half of them are DNA repair genes, and half of them are tumor suppressor genes or chromatin regulation genes. Why is that important? It's because when you have a DNA repair defect, you can have synthetic lethality due to the loss of homologous recombination function. And you can have actually effect from PARP inhibitors in these tumors that you don't have in the wild type tumors. There are some studies looking at this where you stratify regarding your gene mutations and trying different treatments regarding your mutation, like precision medicine. Yeah, I will not. Yeah, so we also looked at the polymorphisms, single nucleotide polymorphisms, with which are can be only one nucleotide difference, but it can make a very big difference in the gene's function. We found that in the literature there are many polymorphisms that have been reported, but as you see, this is in an Australian cohort, and this is in an Italian cohort. And there is no overlap between these two studies, which is a little bit strange. There are also polymorphisms in carcinogen detoxification genes, but they got opposite results in Finland, opposite results in other countries. So I would say that, okay, we have to just go past this, because the conclusion is that there has been no study since 2006 on the polymorphisms for mesothelioma. So that means that there is a problem there. So if we make a summary on the talk today, is that there are three subtypes: epithelioid, sarcomatoid, biphasic. Mesothelioma has a poor survival, 12 to 16 months for epithelioid and 4 to 6 months for the sarcomatoid. 5-year survival is less than 5%. Mesothelioma has high rate of innate and acquired chemoresistance, but there are some long-term survivors. Only one FDA-approved biomarker, mesothelin, which is can be useful for diagnosis and following treatment response in mesothelioma. Surgery is still experimental. Double immunotherapy may be a new standard of therapy. It works equally well for non-epithelioid type. And second-line treatment, I didn't talk about this, but second-line treatment after PEM-CIS can be different things: vinorelbine, immunotherapy, lurbinectedin, but nothing is really clear here. And about genetics and future perspectives, I think that genetic testing of high-risk individuals may facilitate clinical genetic counseling and help us achieve early cancer detection. Mesothelioma patients with germline mutations, they have a longer survival, and this is important information for patients and one reason for germline testing. BAP1 may be a new marker for chemotherapy in mesothelioma. Germline testing in mesothelioma should be mandatory for treatment and prognostic, as well as to uncover familial cancer. It is so common with germline mutations in mesothelioma that we probably should go for germline testing. Identification of inherited mutations and understanding the oncogenesis mechanism may allow us to find potential candidates for targeted therapy, for example, PARP inhibitors, and guide the choice of drug treatment and give mesothelioma patients a better chance of prolonged survival. So here I'll stop to welcome you to our symposium for biomarkers in Trondheim, 14th to 18th of June this year. I will send you an invitation. It's an international symposium, but it will be virtual this year. It will not be in person. Here are collaborators from our university in Norway, in Crete, in Denmark, and here is our affiliations. And this is from our vineyard in Samos, making sweet wine. It's a very nice place. So
thank you.
I will take questions.
Thank you for your nice presentation. We have some question for you from Professor Ülke Yılmaz, who is head of Turkish Respiratory Society. She thanks you for your nice presentation, and her question is, EP plus nivolumab efficiency has been demonstrated in patients with high tumor mutation burden in non-small cell lung cancer. What are the evaluation results of TMB in mesothelioma?
Yeah, it's not a significant relationship. So we cannot use it for as a biomarker for mesothelioma, to my knowledge. Maybe the studies have not been finished. Maybe there are not enough studies, but to date, there is no recommendation for TMB or PD-L1 for mesothelioma, because we don't know the biological basis, or we don't know if it's not a clear relation with response and survival.
We have second one from Dr. Metintaş, Muzaffer Metintaş. I will read his question for you. He thanks you so much. This is very important issue for mesothelioma. If we analyze BAP1 presentation in tumor tissue, is there any importance for determination of chemotherapy response or prognosis analysis?
Yes, thank you. We believe that from our study so far that it's predictive for chemotherapy with the pemetrexed and platinum, because we see that those that lose BAP1, they are BAP1 negative, they have a much better survival on chemotherapy than those who are has not, and it's independent of sarcomatoid epithelioid. So so we are the first to report this correlation, but we need a bigger material to validate, to be sure and say, yes, you can use it for the clinic. But I think it's a good idea to stay in BAP1 to all mesotheliomas, because then we can also do studies with all your patients and compare survival and really see if there is a true connection. And I would say that if you and other centers in Turkey are interested to collaborate on this, we are very interested to take you and work together on this project.
Yeah,
But
yeah.
yes, I think it's a very simple analysis, and it gives a lot of information, because as you see, BAP1 has so many effects. So if you lose BAP1, then you have a problem DNA repair, you have a problem with other things, and this makes the tumor different. It makes it more susceptible to DNA damaging agents like cisplatin and pemetrexed.
Okay, I have a question for you. Do you believe mesothelin to determine chemotherapy response?
Yes, it's. I think mesothelin is a little bit underused. I think that it's better than CAA in colon cancer. I think it's better than CA15-3 in breast cancer. I think it's as good as PSA in prostate cancer. So I think it should be used, but many it is not recommended in the international guidelines. I think it should be a possibility to test it, because it can be very helpful for pleural fluid patients that don't have a diagnosis. It can be good to follow if you don't have a good what shall we say? If you can't see really differences on the CT scan, you can maybe see the difference in mesothelin. And it is, I think that mesothelin also is something that should be used more and also studied more for this use. So, but it's not the international recommendation. And so it is, it's up to you.
Okay, what about the response rate? I mean, percent change or percent on mesothelin for partial response or progression. What about the change of percent mesothelin?
Okay. If you have a normal mesothelin before treatment, then it's not necessary to do. You should not use mesothelin for response. If you have a very high mesothelin before treatment, then you can probably use it for following up the treatment response. But I cannot tell you how much percent change with, for example, stable partial response or complete response. What we know that if you do surgery and you remove all the tumor, mesothelin can become normal. Okay, so in some cases, when you remove the tumor, mesothelin goes down to normal.
We have a thanks for you, Professor Mehmet Karatağ. Thanks you for your nice presentation, and from Professor Binnur Önal. Thanks you so much.
Thank you.
For all these novel data and quite interesting. We might have multidisciplinary collaboration pathology. She's pathologist under the umbrella of Eskişehir Osmangazi University and Mesothelioma Study Group in Turkey. And another thanks from Betül. And yeah. I have another question for you. Many of the targeted therapy did work